Hello, good morning, good evening. Thank you for listening. This is a medical cannabis program and I respect the law and I hope you do too. All this information today is for education and scientific purposes. The information is fact-checked and any papers that are discussed will be put in the description below. And there's no topic today. It's an open topic again. So if you're rewatching this back or re-listening back, please skip forward until you see some slides or till you see the screen change. And that's generally when it will start. It might start a bit before or a bit after that, but that's a good way to pick when to get the action instead of listening to me go on. But today is actually a good topic at the start, and today's show is dedicated to Bryan Cannottell. That's his screen name that he used to go by. Yeah, I'll just leave it with his screen name, but not say his real name just in case. But quite a lot of people know him. He's a really nice, cool, respectful bloke. I don't know how to put it any better. Actually, before we get into it, I should be showing I can share his. Allow. There we go. Look at that. There he is there. I used to get quite happy when I speak to him. For many hours speaking to him. Oh, did something happen? Testing audio. Yeah, audio's working. Yeah, he's just a really nice bloke. Don't know how else to say it. It's a shame. His heart got the better of him. He had a big heart. He never used to yell either. He's had some really cool traits. Just never got real fallen upset. I used to admire that about him. And it was 4 months ago now it happened, but I only just recently found out thanks to his son. So thank you very much for letting me know, and I'll pass this on to the community so they can all think about how cool he was and remember his cool points, which there is many. That's for blooming sure. So there's nothing really just on today, so I'm waiting for usually people get into the chat and they put some discussion topics or ask questions. More so, that's the bit I like, is the questions, because that drives me to try and get an answer for them. And I'll show that all my university slides that I've got, which are recent. They're all within the last 10 years. The whole lot of them, the thousands that I do have, just to try and get the answer. There, you wrapped up. Yes, you remember him. He was a cool bloke, wasn't he? Very cool bloke. I was excited to get a message from him, and I thought, cool, I haven't heard from you in quite a few months. And it was from his son. I went, fair income. I thought, because Anyway, these things happen. Hello, Lene. How are you? Yes, you knew him quite well too. Seems like quite a few people, the cool people in the community, knew him. How could you not? He was just a nice bloke. He had really good knowledge too. But most unfortunate. It sure is. It makes you have to appreciate what we do have and the things we do have around us, and try and reflect on that. Live the now. Try and enjoy what we have. It's very hard because you're always in the past and the present and stuff like that. I'm not going to go on about this because it's supposed to be a medical cannabis show, but I've got to show a bit of respect to the cool people. So, just there's too much. It's too easy to disrespect or to put people down, but draw the positives. We've all got this. All our days are numbered, all of us. So why not try and enjoy them somehow? Anyway, there's 5 minutes. I'm nearly starting to get teary. He was a blooming good bloke, fair dinkum. All right, change the topic here. I'll just show this for, then I'll change the topic. It was always up for a good combo. Yeah, just rad. All right. 5.5 minutes. Now what can we talk about? Now what's happening? Jeanette Graham says, we've chosen few who get to wake up today. Good day, peeps. I hope everybody's well. I'm sorry for your loss. Yes, it's sad. But I like that show, Alaskan Bush People. And one of their kids recently said, oh, that was the father actually that just passed. And he said, try not to mourn me, but try to be happy and respectful that you're still alive. So throw a party that everyone's still alive and try and have that type of fun instead of trying to remember, the sad bit about the passing. And I thought that's pretty cool. So that's what I try and draw on. Try and be happy. Yeah, I can still celebrate that person. Celebrate their good attributes and that types of stuff. There's not many good people around, and he was really one of them. Get me off the topic, please. But yes, we've chosen few who get to wake up today. Oh, talking happened to one of us. We just all appreciate, well, not everybody, but it makes me want to appreciate everyone more. And, it's just full on. Life's too short. Raptor says, how many generations would you need to work with a cultivar to make it stable? It's talking about its heterozygoticness. So they reckon that it's 7, but you can do it sooner. You can do it with tissue culture and a few other selection ways to make it sooner. But in general, they reckon 7. And because the questions aren't flying, let's just have a bit of a squeeze into this. So why is it 7? Or why do they say 7? I'm sharing, yes. Be in breeding section. I need a few charts. Let's just start from the start. So what's the question again? How many generations? Just for stability. Well, they say 7. Oh, we're not going to go this heavily into it. Yes, pure line theory. Johansson's pure line theory. Is that a good way to show it? No. There's a chart that it will come to here, like this, but it's got more years into it. So it just shows how they cross with the parent, you back cross with the parent for stability, and you're increasing it. So actually, I should just go to the 7 one because you can read in here. After you marker-assisted selection from your marker-assisted breeding, you'll go and get your ones that you want and then back breed with the parent. So in other words, save some pollen and then sprinkle on the one you like, get the seeds off that, grow it, sprinkle on pollen again, and keep doing it until you get right down, and then disease screening. So this would have been a good example of how to do it because there was a disease they were trying to get rid of out of the parent. So by putting it through all the time, you'll come and you'll see before it's gone. So that's a good way to tell that that might have only taken 4 generations to make it stable, but in general, it's 7. Actually, let's get to the 7. Oh, here we go. So they were just repeating it just to make it stable. Disease-resistant plants are self-pollinated, harvested separately. Individual plant progenies are grown, and disease-resistant plants similar to that of the parent is the outcome of the 9th year. But see how that they've repeated just for stability, just to back cross this to 10th, 11th, 12th. And then this program has started to, for the commercial sector, they've gone the 10th year, then they've started to reproduce and put them out in the 12th year. No, because we're going to get into these longer charts that I have. Approaches, no. Well, fair income. It's like that. First generation, you select one, and you select the second generation, put it back with the parent. Third generation, put it back with the first parent. Fourth, back with the first parent. Anything here? There's a little slide that I made. No, not really for that question. Explaining genetics through a family tree gummy bears. That's just using the colors. So the offspring is supposed to be 50% of each, and they'll breed back. Actually, no, that's not the example. It'll confuse. Let's see. No. Well, here we go. This might be it. STS. And then what they select: 2, select 3, select 4, clone, seed, and that's the pollen donor for the 5th. 2, 3. It's this first one up here. I'll move the top bit. Oh, it's crossing. That's how we're going to cross. What's the bottom thing? C is crossing. Pollen donor phase. Oh, bad example. That was something out of a study. That's the ratios of, well, the reason why you do the back cross multiple times, this is sort of help to the reason why, is because every time you do it, but the first one you'll do, you'll get this 50% blend of both the parents, and the third one you'll do, the ratio expands heavily, a lot. So to get the traits that you want from the parent put in the offspring, you'll keep crossing it back with the parent and then looking for the offspring. So if you want the green, for instance, here, and that's the trait that you want out of these parents, so you'll be selecting the green one here and putting it back with the parent. And then when it goes again, it'll have a higher ratio of greens or the trait or the gene that you want to be dominant in. So there's 30-odd thousand genes in the plant. So yeah, that's a way to pull it out and use it. What's this about? Heterosis. Oh, it's hybrid big. I thought it said heterozygotic. Horizontal gene transfer. That's implementing bacteria. That shouldn't be there. New breeding techniques, double haploid. And this is the way where you can get your 100%, you don't take 7 years. You can do it through tissue culture. Oh, this one up here, look, traditional breeding methods. This says 8 years. Oh, this is an excellent example. So the parents, then you got a 50% blend, and then you'll go and make a haploid out of it. So you'll get the chromosomes and you'll pull them out and it'll be separate. And then you'll go and double that set of chromosomes because it has one set of chromosomes. So it's not enough to be a diploid. So you'll have to double it and then you put it back through again and it becomes a double haploid. And that's a diploid that has 2 sets of genes in it, and that means it's fine. So you can go through the tissue culture down here. In the first year, you can do it, or up here, you can take it. Generally, they reckon 7 years, but this chart shows 8 years. Actually, what does it read? In the last decades, alternative and more advantageous techniques have been successful. Very good. These techniques based on androgenesis and gymnogenesis allow it to obtain pure double haploid lines by tissue culturing through embryogenesis and callogenesis. Okay. That'll do, through tissue culturing. And it's surprisingly, I'm not going to say easy to do, because you've got to have the right environment and you've got to have the right sterility and the right chemicals. I think this is the one I was after. And it's blurry. Ha! Fair income. Isn't that a shame? Marker-assisted breeding. Well, that's how you do it to select your marker. So your marker would be, you want it to grow with 3 nodes. So it would have a BRC branching gene dominance. So that would be easy to distinguish. So when you go and cross it, you grow the progeny out and you see which one had 3 nodes. Sweet. Cross it back with a parent. And then draw it out again. And then, oh, only half of them come out with 3 nodes. Right, it's not that stable, or hasn't got the gene. Oh, you're not after gene crossing, you're after stability. So that's the difference between heterozygotic and homozygotic. Far out, I was off the chart then. So that's what I should be discussing, not about how to transfer genes. I'm sure I've got some stuff about, yeah, I thought that was pretty intriguing. The genotype. So the phenotype equals the genotype plus the environment. Oh, here we go. Here, this is more onto it. The homozygotic and heterozygotic. So that's stability. So if yours is homozygotic, it means it's quite stable. But heterozygotic means hetero has a few different mixture. So the genes are very mixed and not as stable. So your tendency to, when the plant goes under stress, it's going to want to reproduce itself or throw out the herms. What's this depicting? This is just the pheno swing from a heterozygotic or a homozygotic once you breed back with those. So the homozygotic is supposed to be pretty stable and pretty similar to, so that's why you get many variations. That's the word. Sweet. Rapid screening. More homos. There's got to be more stuff than that. Feeding. I should actually stop sharing, see if that's kind of helped. I've got to be excited about the growing. Raptor, good words. Was it interesting? They were able to stabilize through callus culture. Yes. Quite fast with the haploid cells, because what you've done, you've got your haploid, and then if you, all you do is double it, and it has those genes in it, and it doubles the chromosome set. So, you know, so I'm going to show something about this. I did a whole blooming course on cell biology just for this reason. Chromosome mutations. This goes into a few human ones, but still relevant. I'll try and just get the heterozygotic some monoploids. So we want the diploids. That's what you're trying to get, the end result. So if you have a diploid, you're going to end up with a haploid, because it's half the normal number of chromosomes found in the meiotic cells. So it might, yeah, be a monoploid, but it's not really, because you've turned it from a diploid into a haploid. And then out of that, you want to double that again, make it produce an extra set of chromosomes. So that's when you'll induce it and turn it back into a diploid. That's tetraploid breeding. Polyploids, okay. Yes, and there's a few chemicals you can do with that to induce it. And oreozolan is one of the safer ones. There's one that starts with C, I'm not going to mention, and it's not safe. Don't use that one. So source oreozolan. That might have helped. chromosome mutations. Oh, no, it's not really. That's the different types of mutations you get from the amount of chromosomes that are in there. But we're just looking to double it. So yeah, look at this is an example of, actually I'm not even going to talk about human stuff. Mutations. Red points. So yeah, see, the chromosome has, actually, was a better example of it. Here, one leg. So you've got a diploid, which has two sets, well, two sets, but this is at one set. So once you halve it, it goes into one, and all the genes supposed to be along the chromosome. So by inducing it and doubling it, putting it through mitosis at the right time and holding its spindle growth to make it produce another one, it makes a direct 100% copy of the original one. So therefore it's stable because there's no breeding techniques to exchange genes. It's just duplicated a second. Fantastic, that was well explained. What was this? Here's I got a double bar. Drosophila, that's the fly. They do a lot of fly breeding because it's easy to breed with flies like mice. There you go, that's what I was looking for, this thing before. Oh, and that's actually what happens. It halves into one, and then you want to make it go back into two. Cool. That's what all those big, long, fancy words will say. Would have been good to start with that wrapped up instead of, I think that's it. I think that's it, mate. Yeah. I made my point. Let's hope you've asked. Oh, there's nothing else to ask on. How can I elaborate then? Because 22 minutes, it's made into a show now. So put your questions through, please. Or I'm going to try and elaborate on stability and see if what I've got in homozygotic and heterozygotic genetics. Excuse me. Thanks. I got a year or two. Yeah, try doing both ways, mate. You'd be surprised how, get your laminar flow hood happening, even make it out of a tote. I made it one with glad wrap, cling wrap, and start experimenting, because that's really all you need for the tissue culture section bit. And you get those chemicals and make it happen. That's it. You've already got all the Petris and everything. So I'll try and elaborate. So unless there's more questions coming through on that, I'm going to look for some, I'm already sharing. Mutations, fertilization, seed embryogenesis. Seed fertilization, no. Let's talk about induction. Well, really it's, all right, let's see what sort of paper, advances in plant breeding strategies. Nope. It's a book, not a paper too, far out. Dioecious and monoecious studies, no. Comparative genetics. For flowering, no. Genomics-based taxonomy, no. All the squash preparation. That's another good one. Fewer chromosomes at 100x. Oh yeah, here you go. So this is one way to do it. So you prepare, and well, if you've got a lab microscope, you put it on 100x, you should be able to view your chromosomes, no worries at all. And this is a way to prepare it. Something else good in that I just saw. No. resistance. Oh, it was that thing that I saw. Okay. I'm still looking for double haploid breeding. There's a link there if you'd like to check out combining multiplex gene editing and double haploid technology. But that's not about that's easy to double, but I want to help you to haploid. Here we go. So after you get the tissue sample, you induce it with your oryzolin, because remember the other one that starts with C is poisonous, so very dangerous to work with. And then after that, at the right mitosis cycle, meiotic cycle that it goes and repeats with it and breeds, it doubles its cell, it holds it, and then it produces a double haploid, and then it's stable as. And that's pretty much it. So you think, oh, what? That sounds easy. It sounds easy, yeah, but try to do it. And then you've got to prove that you've got a double haploid. So, no. So once you do that, you want to view them. So that's where the squash preparation technique is one way of doing it under your microscope. It's this one. Here's a paper on the chromosome squash technique. So I'll put this in the description later on. So you can go through this, and it's one way to view your chromosomes under the microscope. Observable chromosomes in the microscope. This is what you'll see. Tells you how to do it, and you just use the safe oryzolin, and that's it. Store it for that little bit of time. Yeah, if you want to get into all this technical stuff, you can, but just step-by-step methods, which looks like it was that slide that I had before. Incubation, and then they go to view it. You can see it. So yeah, anyway, I won't go on about this, but there's your way to view it. Different ways of doing it, and your expected outcomes. Limitations. You don't need that microscope. You can view it through a normal close field lab quality microscope. So I'll leave that one there. So I'll see if there's anything else. Well, that's really it. I think I've covered it. Plant hybrid, no. Advanced in plant breeding, no. No. It'd probably be in tissue culture, actually. I reckon it would be. Let's have a look in tissue culture. TC, tissue culture. Cloning, pollen dihaploidization. Here we go. Oh yeah, I forgot to say you can also do it out of pollen. But it's called dihaploidization. Here you go, some of these will be relevant. So with the pollen grain, it's a haploid embryo. So if you double that and grow it out, you'll have your plant. So see the anther, the male bits. So inside of that's the pollen grains, inside, and you'll get them, put them under the microscope, you'll induce them, the embryos, and they'll double. Oh no, sorry, the embryos, then they're going to grow up, do it a different way. So they've grown it out. So you just put that microspore culture, it's called, and develop embryos, which will, then you'll sway it with your mixture after you've got your embryos, because it'll have to be an even mixture. Then you'll of oxygen and cytokinin, and then you'll sway it and develop the plantlet, and then you've got your haploid plant. So it's infertile, so it can't be done much with. So yeah, that's when you induce it. So there's a few ways to induce it. You can put it, well, it looks like with this way they've done it in the plant. So if you're going to do it with the plant, it's a bit harder because you've got to keep it all moist, but then you could put your chemical, oryzolin, not the one that starts with C, on the tissue, and then that will develop outwards, put it on a meristem, and then that will, as it develops, it should have diploid features. And then you can test it, but that's the way that this diagram shows. Haploid plants, anticulture. There's a little callus produced by an antler. Isn't that rad? So they're undifferentiated cells waiting for some hormones to be given a direction. So like if you give it auxin, this little plant this cell would produce a root, and if you give it cytokinins, it would start producing pericycle cells as the shoot. Eggplant anticulture. So it's that simple. So you've got your little banana, shows you pollen grains inside of it. Develop it, and look, they've just done on the outside. So there has been pollen grains on the outside of this, and they've just grown the callus that way. So I haven't even had to open to get the pollen grains itself. So that's pretty cool. Heterozygotic population. So it shows heaps of different mixtures of everything. So it's very unstable. So the original question was how to get a genetic line stable. So you don't want it like that. And did you know too, under the microscope, that pollen all looks different? These are pollen grains. Isn't that a spin out? And haploid pollen. So that's okay, just a different picture of the same thing we're talking about. They do it with coconut, so they just go with the pollen grain there and develop it, and it slowly grows calluses on it, and then they give it direction. Isolated microspore culture in another plant. It's another different way you can isolate it, as long as it's sterile. Because what happens if it's not sterile? You have to use massive amounts of sterility, because otherwise you're going to be culturing the microbes that are in the air that are going to contaminate it with. Culture brassicas. See, it works with nearly any plant, in other words. So isn't it fascinating? So you just get the right embryogenesis culture, put the pollen grains in your Petri dish, and look, it starts, well, this one would have had an auxin dominant, so it started to root first, and then bang, it's got its meristems in there, so it can go and give direction, and before you know it, it's got chlorophyll and retaining it, it's produced its shoots. Isolated pollen culture. It was first done in 1970, if you really want to know. Pollens can be isolated from anthers by two methods, the float culture or sucrose density gradient centrifugation. So if you haven't got a centrifuge and want to spin them out, use the float culture. Or just isolate them like I've shown in my videos. I've got a couple of videos on breeding. So, yeah, it's easily isolated and you don't have to use any types of methods. You can manually do it. Sucrose one, different pollen at different stages. This is it's going through its cell. Oh, that's not a good zoom. Where it changes, it goes to split the cell. So it has to halve and realign, and then once it's realigned, it moves apart, like this one. And then bang, you've got two cells from one. It's pretty cool. Pollen cell suspension from a nurse culture. I got that exam question wrong. Is there a nurse callus? Yes, there is. Anthers, yep. See the male bits, they look like bananas. Morphological difference between haploid and diploid plants. So you can see the massive difference on the left is the haploid and the diploid on the right. Because it's got this one, the diploid's got twice as many genes to handle situations. And this one would be infertile. I think I've covered this enough. Yes, getting into fine tuning of tissue culture, but this isn't really tissue culture. Microspores. Okay. Oh, here's a good example of how it grows out. We've seen quite a few of them though. This is a bit more detailed. Microgametogenesis. That's the process happening here before it grows out. Development of cabbages. Okay. Oh yeah, that's cool. That's the side of the shapes. That induction ovules of the flowers too. Oh, look, there's the old one taxonomy of the female bits and the male bits. See, they do look, and even when you zoom in under the microscope, they fair dinkum look like bananas. The anthers, the male bits. And to store the pollen, so this is the sepal, the round bit, the balls. So you'd store them in that. So they're not opened, because if they're open, that means they've passed maturity and they're waiting to pop. So you'll store them when they're full and ripe, and you'll be able to tell that from experience, because it takes about 25 odd days for it to grow, for it to mature. And then inside of the anthers or the bananas, you'll cut them open when you want to use one, and then pop, all this massive amounts of pollen grain come out, thousands and thousands, which you'd go and so you cut it, and then that's where you'll get your pollen grains. So you'll pull one of these out, take it into tissue culture, sterilize it, take it into tissue culture, and then cut the top of it over your Petri dish. Bang, you've just gone and fertilized your Petri dish with your embryogenesis culture mix in it. Shut the lid up and bang, and then you've done it. And then you should have haploids out of that. And you might have to, depends on your mix, you might have to rechange the auxin or cytokinin ratio to get it to do what you want, because it might just sit there and grow in a callus. There you go. And if you want to store these, store them in their sepals. So the millions of pollen grains inside of one sepal can be stored successfully inside the anthers, which is then in turn stored inside the sepal. And you store it in the freezer for long term, or fridge if you're going to use it, because the freezer, it might develop water around the outside of it, which will make it not viable. So you really, to go in the freezer, you want to do it increments, not flash frozen. Oh, and that's a good light thing. Stages of regeneration. No, yeah, the different lights, isn't that cool, to get different growth from different wavelengths, stages. Tetraploid, no. Di-haploid. So that's one they've induced. So a di-haploid is one with two sets of chromosomes, because it was a haploid and now you've gone and put two into it. So it's a di-haploid. So it's like an artificial version of our diploids. So if you were to make one yourself, that's what you'd have at the end, a di-haploid, not a diploid. Which theoretically, it's the same thing, still got two sets of chromosomes, but it was arrived at the two sets of chromosomes differently. That's why it's a different name. Sugar males. No, this is more getting into the polyploids, seeing different sets of chromosomes doing the different, having more benefits in the plants as you increase the amount of chromosomes, because they got more sets of genes to do stuff. Oh, no. Went through that one. That's it. Let's see if there's any questions now. 40 minutes. Not bad, not bad. Raptor says, I made a laminar flow hood out of plastic taupe, a filter, and variable fan. Nice work. Hope you got your HEPA filter. I got a cheap one from one of the major retail stores, and it was like 30 or 40 bucks, and it was double filtered and it had a HEPA in it, and they sold $5 replacements. So I got quite a few of them. So that's what I use in my little tissue culture laminar flow hood. So it's always going to be sterile and clean. And what else are you missing? Oh, a little Bunsen burner, or I use a candle, because that's where you'll clean, because right next to the candle you've got really hot, the microbes can't survive next to it, so that's how you get your sterile, your tweezers and your equipment that you want to use. You need to use it other than fungi. Yes, you're a bit of a fungi head. Well, I meant that in a nice way. You know heaps about fungi, that is. It's small, but I have so much more space. Nice. And nice, another reason to get a microscope. Yeah. With the microscopes, it's about 500 to get your good lab one, and then maybe another 400 to get you a high resolution, what's mine, 16 megapixel camera. They're probably heaps cheaper now. Maybe drop a couple $100 off that, but that's what I got, and it can do the things that I need it to do. Is there anything else that you want me to discuss? What else can I talk about? Sort of got a little bit of time. Did the internet just go off? No, there it is. Okay. What else can I talk about? Well, we were double haploids in tissue culture. The flow hoods. Keep it on the original question, which was, just having a look down. How many generations? Oh, to stabilize. Oh, well, I've kind of been through the two ways that I know, through tissue culture and through back crossing with the parent. Okay, I don't know how to elaborate any more on that without making it boring. A laminar flow hood. Microscopes. I don't know what else to discuss. Help me, people. Give me direction, please. Let's see if I've got anything else in the genetics. Even actually, I probably nearly covered it all. Holy moly. Bit of an update on the microscopes. Raptor says you can get a lab grade microscope now with camera attachment for 300 on Black Friday specials. Check though, mate, don't get the camera attachment, because I bought my camera. I know the place you'd be going to, because there's not many people that sell the microscopes, and that one there in the States, they've got not very good cameras. So I got mine on another website, because after looking into it, I wasn't happy with, I think, the 8 megapixel or something they give you. Like, it's an old school camera. It's one that's not new anyway. 8 megapixel. It's something like that. So in other words, when you zoom in, you're going to struggle. So I got one it's 16 megapixel. Yeah. And it costs about the same as the microscope. But I'm so glad that I did get it. So glad. Because oh yes, you need to be careful with those. Yeah. They got the brilliant microscopes. So if you're not interested in using it with the video, which, well, maybe if you can't afford it, buy the microscope first and then save up next for the video. And I've got a binocular microscope, and I pull out one of the eyepieces and put my video in it. So I haven't got the tris or the monos. So then I can still use one eyepiece, and then the camera's in the other. And to be honest, I don't really use the eyepiece at all now. I just use the camera because I want to see it on the screen. But the benefit of having your microscope on the screen in video format is you can measure stuff. They got these little calibration rulers that you calibrate your microscope with. So you'll put your little infinite ruler, you can't really see, but you put it on the microscope and it shows lines. You measure the width and it shows it should be 0.0001 thingo, and then you'll calibrate that with your microscope and it measures it in pixels. So it gives you the approximate pixel resolution for what you're telling it for the information you put in. And then you can measure stuff really accurately. Then you'll go back on their scale and you'll measure it again and see if it equals what this says on the scale. And it does. Well, if you do it right, it will. So therefore, you can put anything under and measure its width. In like micron, that's how you know of like a trichome head is 20 micron across, or up to 200. But so yeah, you need to be careful with those. I just want one. Raptor says, I just want one that Sunshine. Put your questions in, Sunshine. Just about to leave. Raptor says, I just want one that can connect to my computer. I'll feed it to a large monitor. Ah. If you struggle, send me a message and I'll try and find where I got my one from. Because that was a few years ago, remember? You were around then. You remember when I got it? Actually, it was many years ago now. That's when I'll get into meristem culture. Oh, yeah. Yes, that's so important for the... Well, with the meristem cultures, actually, I'm not going to talk about other people. There's Sunshine. Hello, Sunshine. Nice to see you. How you going? Do you have any questions today, Sunshine? What you missed is on genetic stability. So the question was, how do you make a cultivar? How many generations do you need to work a cultivar to make it stable? So we're just talking about the 7 or 8 back crossing ones, or the tissue culture ways to do it, which you can do it in 1 year. But it's, yeah, you need the right setup. But just fun stuff. And yes, all the Australians are rocking up. Hello, Blinky. That's funny. Some Australian thing must have finished. We're talking about genetic stability and tissue culture and back crossing. So if you have anything around that, please put it in, or actually in general, you can ask any question. That's what it's about. But that's what we've been mainly talking about. Yes, I forgot what I was said then. So it's 47 minutes. And I've covered those 2 points pretty decently on how to go about it and how to test your results. So I don't think I can really elaborate any more on it without boring. Because yeah, I went through all of those. What's this? Oh, here's a link to do with chromosome counts. All right, I'll show it. We're not really talking about much else. No. Well, it is to do with chromosomes, because you know the amount of chromosomes is the ploid. Monoploid 1, diploid 2, tetra 3. Oh, triploid, sorry, 3, tri is 3, tetra is 4 in Latin. So that's the amount of chromosomes that are in it. But what's the point in showing this? Induced autoploids. This is the different ways you can cross with an autoploid. There's 2 different ways you can breed, through autos or allos, or you can do segmental. But the different ways are fascinating. And a lot of tests are only being done one way. And I've done both ways and found that you can get hybrid vigor by doing it a different way. Anyway, I'm trying to relate this to today's show. Alloploids, synthesized allos. This is different ways to breed to get your triploids and tetraploids. It's not to do with haploids, which is really what we're talking about don't know if I should put that one in. I suppose it gives an example. All right, I'll put in haploid breeding and see what comes up. Double haploids, been through that. I was hoping some papers or something might come up. Production of it, no, mapping. I think I've told the two different ways and how to test. I don't think I can elaborate anymore. I thought that was something there, though, a study or something on these chromosomes, pollen culture, sterilization, microbeads, meristem culturing, no cloning, no. Is there anything else in here that's relevant to haploid production? There's a few studies. Dissection of meristems, no. Plant tissue culture, no. I'm sure there'll be some. Preparation, no. Oh, that's it. Back to this one. I don't know what else I can share with. G'day, your keyboard battery's low. That's no good. Keyboard battery's low, so I missed the, and it ended. Looks like it just ran out. Oh well, that's okay. You can save your question till next week, sunshine. But if anyone's got any questions, please put them in chat now, because we're just about to leave. And today's show will be retitled on genetic stabilization, the two ways, tissue culture, testing of double haploids. That's a big word. Testing of chromosomes. MO, that'll do. Testing of chromosomes. Oh, it's with a light field microscope. Oh, I'm not going to put all that in. Genetic stabilization, two ways, tissue culture, testing of chromosomes. What else did we discuss? I think that's... Oh, hang on, there's a question. Okay, here's a question. If you made a new living soil, what's the fastest way to inoculate with a diverse microbial profile? You want to give the microbes what they want. So microbe needs things to survive, so you'll be trying to give it the most amount of that. So what's the fastest way to load up with diverse microbes? Well, diverse is exactly the word you said. So you want to get different types of microbes. Oh, wow. Blinky, thank you very much for that. Look at that, everyone. Thank you. You must like the double haploid production, huh? Good on you. Thank you for that. Very appreciated. Essentially, the fastest way to load, yes, diverse microbes. So you want to get a source of different microbes. So where are heaps of healthy, beneficial type microbes where you can buy them, and in Australia it's top dollar at the moment, it is. Or manure is a good source of it. So make sure that the being that you're getting it out of is healthy. Like horses, for instance, it will have antibiotics in it, so that's not good. Humans will have all sorts of fats and oils and probably other tablets that they've been taking, so that's not good. So something that eats grass, like kangaroos, rabbits, chooks, those types of things are always, you sort of can't go wrong with them. Cows, it's a larger amount. The older pats have a lot, because 99% of the microbes haven't been cultured yet because they can't test them in labs. So who's to know if they're going to form spores or not? So if you really want a culture to amplify the microbes, what I do is what I have done is I use cow manure that's a bit wet. So it won't have had a chance to dry, and all the spores inside of it won't have had a chance to possibly die, because some of them mightn't sporulate, or they mightn't form spores or cysts after they die for them to reproduce. Bacillus thuringiensis, I think, is one of those types, because I've got to re-inoculate that every 3 months for that reason. So back to the question, what's the fastest way to load up with diverse? So if I was to do that, I'd be sourcing wet cow poo. I'd make a tea out of it, a manure tea. So you'd want to bubble it for a minimum of 24 hours, because that puts enough oxygen in where it kills all the anaerobes or the pathogenic microbes. So I think it's 24. I always go about 30 hours. And you can see from that growth chart the growth curve from microbes, that longer than, it goes through 4 different cycles: a log, lag, a stationary, and then death. So you want to pull it out when it's producing. So when it's at its stationary at the most, or when it's at its death phase, is when it's dropping its numbers. So you don't want to culture it. You don't want to be using it then, because the numbers have gone down. So around about 28, 30 hours after it starts, that's generally a good time. Gee, it's hard to say, but that's generally from my experience and from the charts that I've used with my particular microbes that I've done. There's some more questions. Oh my goodness, down here. There's add-ons to that. Okay, well, I'll fix that one. Raptor says, inoculate with compost tea for a quality compost source. Yes. Yep. So another source is, like Raptor says, the compost. So you can go and source that. Manure, anything breaking down and rich in, it's hard to get rich in CO2 to know that source. But you don't want it to be, if you dig down in under a forest, it might smell bad, and that's not good, because that's not the microbes that you want. But yeah, compost tea is another way. Vermicastings has stacks in it, so you can tip if you haven't got access to compost tea, you might have vermicastings, or a major supplier at the shop has vermicastings that you could probably use, which would do a similar thing. Oh, here we go. He says, rotting wood, fungus, swamp water. Okay, well, rotting wood has only particular microbes in it, and it might just have fungus in it. So I wouldn't expect to get much out of it. Because what's the area where bacteria multiply? So it wants to be very wet, above 91% moisture, very wet in other words, and in elements that they can break down to exchange. So oxygen, innate environment, and I'm getting leading off track here a bit. I'll go back to Sunshine's question. Is fungus good? Well, fungus, compost, mushroom compost, you might mean to say, or just what, fungi? Well, fungi, if you get a mushroom and want to culture that, there's not much bacteria on it. So next one, swamp water. Swamp water, it's very stationary, so it might have a lot of anaerobic pathogens that have multiplied in that. So that's one you'd have to particularly watch, especially if there's any smell to it. But the edges where the swamp water is, where it's a bit darker, whereas a lot of humic substances have been built up, that's probably a better area. And you skim off the top, you wouldn't dig down deep. Oh, fish pond water. Well, fish pond water's got to have a lot of nitrite in it that's been broken down, so it might have actually something negative in it, which is nitrosomus, which volatiles off your nitrate, turns it back into dinitrogen and gets rid of it. So fish pond water, I wouldn't expect, Well, it depends on the health of the pond. All these sources that you say are very good. So you could just even use a little bit of each one, mate. But I'm just thinking of the one with the most enriched area. You saw from a local park? No, because idiots will be tipping all of their substances into it, so you don't know if it's an organic source. 36 hours is optional, optimal. I think you're making a statement. Do it whatever you want. It's all to do with the growth curves. So if you want the most amount of numbers in there, you want to make sure it's in. Yeah, I'll show you. You've asked that twice. Microbes. Now, where is growth curves? Here we go. Growth curves. Oh, that's the temperature growth curve. Okay, that's not really helping. Well, you know about, you want to maintain the right temperature. That's because we have mesophilic microbes. So if you go and get them from hot sources, that's not going to be good for our regions, because in pots we're looking at the 20 to 30 degree to 35 degree mark. So culturing other types of thermophiles or psychrophiles, which are even colder, it's not going to help. I'm getting off the topic. I have to stick to the topic, so I don't want to elaborate heaps. Microbe. No, I did an assignment on it. A rad assignment. That's what I'm looking for. I hope it's here. It's just one graph. It was a month-long one. I tested. There it is. Here we go. So this is the phases of the microbe. So lag, log, stationary, and death phase. And that's an approximate across the bottom with a culture that hasn't been really enriched. It's the viable cells on the side, on the y-axis here. Optical, that's how a different way you can test it with your turbidity meter. But so you want to be harvesting in here to here, from there to there. So in other words, you want to take it out of the bubbles and tip it into your substrate at that time. So I try and pick this time because at least it's finished all of its multiplying. So the number of the cells has gone up from the time it's gone in, and this is my results from. So all of these are compost teas, I think. And this one's root tea. So you can see the root tea goes up and keeps going up because there is so much beneficials around the roots in rhizosphere and the rhizoplane that have been multiplied. So they've got out into the water. Cool, that's what they want. They found oxygen, bang. They only need one more thing, their carbon source, and they can start to multiply. So they found that from my sucrose or what do I use? What's that? Molasses, sulfur excluded molasses. And at 12 hours, it's gone up. And you can see it's gone stationary for this long. And now I would think I'm going to harvest after 36 hours. But it's climbed again. It's gone through a different phase again. And that's very interesting. That's an uncharacteristic curve. So something here must have happened. Possibly I put something else in, which I didn't. But the normal curves are up here. So you can see they're kind of going through this phase over here. Lag, log, stationary, death. Where it goes up a bit, stationary, and then it just stays the same. So they're similar. But that was after many different teas and many different conditions, many inoculants I put in over a month just to see the duration. So you can see here, it all starts to dip, goes down after 36, except for the root tea. So that's why I'll only go for about the 28 to 30 hour mark, or even look up here at 20 hours. It's generally not too bad. So 12 to 16, but that just shows you that question for 36 hours is optimal. I hope that helps for that. Raptor says, yes, manures from herbivores is ideal. My go-to is Malibu compost, which is made from dairy cow manure. Yeah, Raptor. Good info, thank you. Thanks for the words. Obviously, any aerobic sources, never anything will smell. That's right, yes. Because it's generally not bad, and bad things have the bad aroma to it. Good smell is supposed to be the geosmin, which is a smell of good earth. Worm castings are great as well, says Raptor. Thank you. What if you have a pure, clean forest soil? What if you have pure, clean forest soil? It won't be that clean once you look under a microscope. It'll be teeming with microbes. But it's just another way of getting different microbes, because different microbes do different things. So by having different sources, you're mixing different cultures in, and who knows if they're symbiotic or if they're mutual, or if they're bad predators or pathogenic ones. So you don't really know until you establish the food soil web. So Sunshine's trying to create it, and he's looking at different sources to get his microbes happening. And it's a great question. You can, if I was set up, I would tell you how to get them off me, but I'm in the process of setting up to get, I got microbes that produce hormones as well as nitrogen, phosphorus, breakdown, all that sort of stuff. And I'm finding it blooming amazing, but I haven't successfully got to commercial yet. But what if you have can you elaborate on that question, please, Blinky? What if you have a clean, pure forest soil? It's still another source. So whack it into your teas. If you want to multiply it fast, you put it into a tea with a bit of sweetener for the carbon source, sugar, sucrose. How much would you put in? For 20 litres, for a 15 litre or 3 gallon drum, you might put in a teaspoon of sugar or your carbon source. What if you have a clean, pure forest? It's still another source. It won't be clean because it's not tissue culture set up, so it'll still have microbes there. It just won't have maybe as much as you'd like if it's a clean forest. But they're still there because nutrients to cycle, they need microbes and fungi to have the cycling process. So that mightn't just be the numbers that you want. Can you elaborate more on that question if I didn't help, please, Blinky? Sunshine, I think KNF farming, I imagine, would be ultimate. Are you kidding? They use lactose, mate. Lactose is a biofilm producing genus that isn't symbiotic. So that's why I've never really understood much about the KNF, how people have good results. It might be that lacto that's actually brilliant. But I've never gotten into it because I learnt that in my microbiology stuff. Here's Yield Source. How you going, Yield Source? Nice to see you, mate. I saw that Pollen Sachs had his luck. That was very nice. I commented on his video. Thanks for that info. Sunshine, if the temperature is warm, 24 hours would be fine. It depends, mate. It depends if you're on your enrichment culture. Well, remember that chart I showed you before? Some of those lines went up and kept going up. So it really depends. But if the temperature is warm, 24 hours will be fine. I wouldn't risk it. I'd go always 28 to 30 hours because you don't know if you've got a pathogen that's a bit resilient. It could be a, what's it called? There's facultative and obligatory. It could be a faculty anaerobe, which means that it's not obligated to use non-oxygen. It can use oxygen, so you don't know if that microbe might exist. That's what that type of microbe is called. G'day, g'day. Sunshine says, take established healthy forest soil and bubble with molasses for 36 hours would go well. Well, it's another source to get your microbes. So that would be one way. You could do it with that, bang, done. Then you could do it another way, the same way with manure, wet manure and dry manure, and all these other sources that we've talked about, the vermicastings, compost. Sunshine says, interesting. I assumed IMOS were the benchmark of microbes. The more you know. What's IMOS mean? Please, Sunshine. I don't understand. But yeah, you know, an example of this is in the largest pumpkins in the world grow in Anchorage, just out of Anchorage in Alaska. When they did a soil sample there, they did a metagenomic study of the microbes in the soil, which the metagenomic means, meta means lots, so they studied the lot of the microbes in there. And coming back, they found that there was about 20,000 different species. And people might have 6,000 to 8,000, and they're doing really, really well. But they found this well-established ground had 20-odd thousand that were working with each other, and that's why it went so swimmingly. Yeah. So it's microbes, they bloom help. IMO is Korean natural farming technique. Oh, okay. Remember, there's many ways to grow, many ways. And the plant only needs 1.5, it gets 95% of its nutrition or elements from the atmosphere. So 5% only have to be given to it. And how you get that 5%, it can be in many different ways, many ways. And then you've even got cultivars that are used to starving, so they're used to low amounts of that. They can be nitrogen deficient, still reach their genetic, or not genetic potential, but enough to successfully complete their life cycle. So there's just bulk ways to grow to get that result, mate. Some IMOs take 6 months to develop. What's an IMO? Oh, some Korean natural farming techniques take 6 months to develop. Okay. See, I'm not really into that. I'm into diversity of microbes and getting the right ones to work for me, especially the ones that produce gibberellic acid in flowering. Oh my. The same results as CO2 enriched environment is amazing, just from microbes. So yeah, microbes, the right ones work in the right environments, and like that Korean natural farming, they use their lactose and love them and thrive. Where me, I don't use the lactose. I use other genuses that are beneficial. I don't want to go into microbes. I can talk for another hour. Please put your questions in chat if Blinky didn't add to that. What if you had pure clean soil? You could still make a culture. Oh, I think that's right. So you must have clean soil around near you. Well, that's one source. So you get it from the forest, and then you go dig up next to a tree, which has been dropping in the open field. You'll see a big massive tree that's dropped all its leaves. So you might even look on the maps satellite from your local area and make it high res, and then you can see where it's green. So you're looking above and looking down, and you'll see around big large trees are green. They just harbor fertility. So that's the areas of soil that I would be focusing on. So you might get some from the forest, you might go some and look for that green things. Then you might go down to the lake edge and maybe take a few scoops, or down to a river edge, or your vermicastings, compost heaps, different manures that are organically sourced. So any types of these things have microbes in it that you can use. And who's to know, are they going to be beneficial? So that's when you have to mix them together and see what happens. And you can be the test of that, because that's the hard bit. Otherwise, everyone would be using this one blend. So, yeah. But knowing how minerals cycle, either you can give them a mineral, which is a nitrogen element, so synthetics, or you can give them immobilized nitrogen elements, which are bound up inside organic matter, like grass has heaps of nitrogen. And for that to be unlocked, microbes have to unlock it. So it comes back to two sources. You can give it minerals, synthetics, or you can give it organic matter, where microbes have to break down that stuff to make the elements available to the plants. And I massively support the second one, which is sustainable for the earth, and the way Mother Nature has worked for 4.6 billion blooming years. Actually, she hasn't been cycling for that long, but that's just the way to sustainable. Where especially Australia, there's not many people from Australia here on this that listen to this stream, but farmers in Australia have severely damaged the earth, and because of the organics is new to Australia, so I've been told. That's the agricultural suppliers have told me this. Organics is new to Australia. Yeah, so that tells you how everybody here has been educated and how they conduct their farming and gardening techniques. It's blooming sad. But it's only to do, it's just education, that's all. And marketing. So not much money, or very little, can be made out of synthetic type of a grow, because you don't need pesticides or herbicides or anything like that. You don't have to buy ongoing amounts. And I'm just preaching to the converted here. Put your questions in, or your statements, because I'm going to wrap it up, I think. Yes. Because I don't want to talk too much about the microbes, because I can really elaborate, because that's, I believe in. I've seen the results. I know about the processes and the chemical conversions, about nutrient cycling. It's it's really cool. I'm not sure on which microbes, but I know that particular ones do particular jobs, which can be beneficial. You're welcome, Wrapped Up. Always, I appreciate your help too, mate. Your words are make the show as well. Thank you back. Same as everybody else who contributes in chat. I really appreciate that. And as well as people like Blinky too, who contribute with little bonuses. That's rad. But everybody just helps make the show. Otherwise, I wouldn't be talking. So I really appreciate everybody who's written in. And even if you're a bad person, still write in. Just be polite and you'll be accepted. That's all it is. And even if, because there's quite a few people in the Australian community who aren't the best. So I don't care about if you're not the best or the worst, or if you've done this or that. You're still welcome. Just be polite. And yeah, that's about it. This show's for everybody. I want to help. All that want to be helped, especially in the organic sector. Australia needs so much help. Agricultural suppliers even saying the wrong things. And they're the people that supply the farmers, and they're supposed to be agronomists. Don't get me started. Anyway, thanks everybody for rocking up and listening. Your ears were appreciated and your words were respected. Goes out to Bryan Cannottell. Ron, peace with you. And I'll be doing this next week, same time. So save your questions for then. If you have some, I'll be happy to elaborate on them and make a stream out of it. So thank you everybody for listening again. And happy breeding, happy growing, and good health to you all. Bye-bye. Thank you. Thank you. Thank you again.